Okay, so every time a cell divides, it needs to completely duplicate its whole set of DNA, which is 6 billion base pairs long, so, I mean, that's a lot. So stemming to conservative replication, basically the DNA strands separate, and the way that they do this is the enzyme DNA helicase comes, and what it does actually is it binds to one strand and basically works its way up, and it breaks all the hydrogen bonds between the bases. What would happen is that your free nucleotides, which are complementary, would hydrogen bond, and then you'd expect DNA polymerase to just come along and join up the phosphate sugar backbone. And the thing is, DNA polymerase can't start on nothing. It has to be bound to something already for it to be able to duplicate. So it can't just get two nucleotides and join them together. It has to have something to start on. So if we move helicase up a bit, so we have a DNA polymerase for each chain, and we have an RNA primase. An RNA primase can synthesize two RNA nucleotides together and forms a little sort of primer that binds here, and then DNA polymerase can bind on and just do the rest in DNA. So primase will stick an RNA nucleotide here. So DNA polymerase can bind onto the primer. I mean, the primers are like 2 to 10 nucleotides long. I just didn't have enough space. It bind on here and it would join up all these sugar phosphate backbones. And do you remember I said that DNA has a polarity in that one side you've got a 3' oxygen, so you've got your hydroxy group here, and the other side you've got one here. Well, it turns out that DNA polymerase can only add new nucleotides to this oxygen. So the chain is always going to grow from the 5' end to the 3' end, so they're always adding nucleotides this way. So for these ones, that's the shape that they are, that's fine. DNA polymerase can just go up and add new nucleotides. But on this side, the new chain is being synthesized this way. And because DNA polymerase can't go in this direction, it has to add to this group. So it has to grow in this direction, 5' to 3'. We have a problem because the DNA polymerase moves up in this direction, but it has to be synthesizing in this direction. So the way that it does this is it does this in little bits. So it's really very cool. And primase will bind maybe in here. The primase will do that. And then your other DNA polymerase, it will be here and it will work its way down, and then it'll be back up. And then primase will get another nucleotide in over here, and then the DNA will go and synthesize this way. So it does it in short strands. This is looking very messy. So this strand, primase will add a primer here, and this will be 3' and this will be 5'. And the strand will grow in this direction. And DNA polymerase catalyzes the condensation reactions between the adjacent nucleotides to form the phosphodiester backbone. So that can go like that. This end is 5' and this end is 3'. So the DNA that's being synthesized, 5' is this way and 3' is this way. So these are your primers. So so the primase or enzyme will put a primer every so often, and then your DNA polymerase goes this way. And these little bits are called Okazaki fragments. So this is the leading strand, because it's really easy. And this is the lagging strand, because it has to be synthesized in little bits. And then at the end you get another DNA polymerase, and it comes along and it chops out all the bits of RNA and sticks back in DNA. So it's very clever. And just to remind you why it all works, because of complementary base pairing with adenine and thymine and guanine and cytosine. So that's how it all works. Because of complementary base pairings, as soon as DNA is pulled apart, as long as it's held apart and free nucleotides are available, they will always bind with their complementary base pairs, hydrogen bond, and then your DNA polymerase just catalyzes the condensation reaction. Actually really hard to undo a double helix, because as you take apart the strands, these bits become more tightly coiled. It's called supercoiling. Okay, so what we have is these enzymes called topoisomerases. We have our double-stranded helix, and say it becomes really tightly coiled, what a topoisomerase does is it will bind to one of the strands, break that strand, only one of them, and then just hold on to one side. Then that strand will unwind, and then the topoisomerase will seal back up the thing. So it means that when DNA gets really tightly coiled, where the helicase is, above that will be really tightly coiled, topoisomerases will bind, break one of the chains, let it unravel, put it back together, and that means that all the DNA is nice and sort of relaxed, as it's called. And yeah.